today I'm going to talk about the work in my lab and also collaborators' lab in designing artificial metalloenzymes for sustainable energy catalysis. As you all know that there are major challenges for sustainable energy, and here's just one example. Let me bring up the pointer. So this is one example of the fuel cells that we've been working on for some years now. And as you know that there's an anode side, there's a cathode side, and particularly for the oxygen cathode side, the oxygen reduction is one of the major challenges because it's less efficient than the hydrogen side. And currently, the state of art is still using the precious metal ion like a platinum, and also, generally speaking, there's still quite a bit over potential, about 350 millimole over potentials. Interestingly, enzymes has been utilized to catalyze the same reaction. In fact, it's a very important reaction called respiration. It's a cytochrome C oxidase. It's a terminal oxidase that catalyzes the reaction to convert energy we breathe in from air in breathe in oxygen to reduce into water, so that it actually convert that energy in nature for so-called ATP is a currency of energy for almost all the biological processes. But these happen to be the same reaction we utilize it for the OR reaction for fuel cells. And enzyme, on the other hand, utilizing the iron and copper. But more importantly, for many years, it has been difficult to compare between the two systems. One is a heterogeneous a platinum system for OR reaction. Another is a homogeneous solution in water. It has been quite relatively difficult to compare the two. And this is one of the paper who collect all the information from the literature in terms of experimental data, but more importantly, using some computational model to really extrapolate the two systems into the one conditions. So this is both the combination of the experimental data and computer modeling to show that for cytochrome C oxidase, there are two models. Either model has much lower over potential. This is the zero over potential for this particular reaction. And then relatively speaking to the state of art, at least in 2010, is either model is better than the state of art, the chemical system models. So they use much lower over potential to catalyze the reaction. This is very important because for energy purposes, we want to put a minimum energy to get a maximum energy out. So this is just one of the many examples that metalloenzyme like cytochrome C oxidase and also hydrogenase for the hydrogen side and in the for sustainable or renewable energies. So these are kind of a metalloenzyme that interest to my lab and many other labs. For example, lignin peroxidase, manganese peroxidase involved in the lignin degradation is the second most abundant polymers. And of course, those system is also very efficient. And also the reverse of the OR reaction is water oxidation and also utilized by a metalloenzymes. So again, for many, if not all the important for sustainable energy are metalloenzyme, that's a great news. But unfortunately for human beings, the nature designed this one for their own goal, for their own kind of metabolic processes. They're not actually, we cannot really easily adapt it for our purposes. They're quite large, they're not very stable and difficult to make. So therefore, for example, the cytochrome C oxidase is a good example because it consists of heme and copper center here working together to catalyze a very efficient OR reaction, so that, and also using iron copper with low over potentials, but yet they have very large protein, contain many subunits, and it's also membrane bound protein. It's difficult to work with and it's very difficult to apply. And so this actually, this cytochrome C oxidase is in the class of the heme copper oxidase because it contains heme copper center. Is actually a very important class of enzyme in biology. They call it heteronuclear metalloenzyme in multi-electron, multi-proton processes. As you know, the OR reaction requires 4 protons and 4 electrons to do the reactions. And interestingly, if you replace a copper center in heme copper oxidase with a non-heme iron center here, it does a 2 electron, 2 proton reaction of NO to N2O. And more interestingly, if you replace the either copper non-heme iron with a iron sulfur cluster next to the heme center, it does this very interesting and very complex 6 electron, 8 proton reduction sulfite to to H2S's. So basically, these hydrogen liquid metalloenzymes in multi-electron, multi-proton processes are very important in biology, but also important for us to understanding, for example, nitrogen cycles and sulfur cycles. And their structure much more difficult to understand and mimic than many of the mononuclear and homonuclear metalloenzymes. In fact, until 2018, and there are no report of inorganic catalysts that all artificial enzymes to really do this reaction. And because it's a quite complex reaction, part of also reason sulfite is very difficult to be bind by any metal ion because it's a very, very so-called greasy, so have very weak binding site in there. So this is the kind of a program we have in the lab for some years now to really trying to understand how nature works and try to make artificial enzymes to understand it and more importantly make something that useful. Obviously, we're not the first to do that. In fact, since 1990s, a lot of groups have been able to make a so-called synthetic models using organic molecules to really to mimic heme copper centers or heme non-heme iron other centers, and also even iron sulfur cluster, heme iron sulfur cluster centers by many pioneers like the Combe lab, Jim Coleman's lab, and then Ken Carlin's lab, and also many around the world. So many of them have been made. They have contributed greatly to the understanding how the native enzyme structurally, how it works, but very few of them can perform the complete for the OR reaction, for electron, for proton catalysis, and also for many of model, they have a poor activity. If they do, the activity is very, very low. So we are actually, when we entered the field, we asked a very naive question. Why don't we see a more of the structure and function model with high activity? So our hypothesis built upon this the following kind of a principle. We could think only by synthesizing the biomimetic model with as high activity as a native enzyme, we can truly capture how native enzyme works, and also to how to do in terms of efficient catalysis. So I like to give an analogy. This is the one I usually give an audience to outside people outside the field. Is that human beings always like to study nature, like birds, for the reason of how birds fly. For us, how native enzyme work in the catalysis. So you can actually make a mimic structurally to the nature in the enzymes as much as you want, even as close as you want. But if you only stay at the structure model side and without function or activity, we still don't know how birds fly. Only one actually makes something that doesn't look like nature structurally, but more importantly, functionally as high activity as a native enzyme, we can truly capture how nature flies. In this case, is how catalysis works. So that's really what we strive to do, is not only make something similar structurally, but also something that's functional. So the question is, again, why, despite all the effort, most of the synthetic model work or even artificial enzyme work only stay at the low level, activity level, or no activities? And so let's examine the active site of metalloenzymes. So they always bury in the middle of the site so that they can actually control the cell activities. And so this is the heme copper centers. And the prevailing view for many years is that they're surrounded by, in the sense, kind of a molecule or amino acid, mostly organic, hydrophobic residues. So one can replace it with organic solvent. So it has been prevailing view. Therefore, most of the synthetic model was done by synthesizing these active sites, putting organic solvent and study, and then try to tune the activities. So is that really true? So if you talk to biologists or particular structural biologists, it's actually you find out that actually for this particular case in cytochrome C oxidase, actually about up to 40% of them are hydrophilic residues, not hydrophobic residues. So you cannot simply replace it with organic solvent. In fact, there are actually a lot of positively charged residues, negatively charged residues. But more importantly, remember from a lot of these enzymes we're interested in, for actually majority of them, actually it's a multi-proton, multi-electron processes. So therefore, proton and also involving water plays a key role. So I display here that all the water has been discovered by structural biologists in cytochrome C oxidase, that this is OR reactions. You can see strategically placed water plays a key role in order to control not only electron transfer, but also proton transfer. So therefore, the true picture of this one is no longer just organic environment, it's actually much more complex. So the working hypothesis when we started working this one, in order to get something very high activity, is that the active site or metal binding site is in a much more complex protein environment. It contains multiple different non-covalent and asymmetric second torsion spheres. And the role of this so-called heterogeneous environment had not been fully addressed by synthetic modeling or artificial enzyme design studies. So we think that may be one of the reasons we cannot get structure and function model with high activities. Obviously, this hypothesis is not new. Most biologists knows it, many chemists knows it. The only question is that which one? We know not every single residue, like 200 amino acids, are all important, right? And actually 1,000 amino acids. So not every single amino acid residue is important. The question is, what is the minimum requirement? Which one's important? What's the minimum requirement for incorporate so it actually can make artificial enzyme to be as good or as high activity as native enzymes? So that's really kind of the goal in our research is to find the minimum amount of information, constructor features to get a maximum amount of activity. So one working hypothesis for us, another working hypothesis that we have is that by looking at biology utilizing the same metal center existing in heme proteins, and they can catalyze wide variety of reactions. For example, the heme protein can catalyze like myoglobin, hemoglobin can do oxygen storage, oxygen transfer, and also we can actually do catalysis, catalase, I should say, peroxidases, oxidase, as I mentioned a moment ago, even all the way to CH1 activation and many other functions. So our hypothesis that since nature using the same heme center, can we take the smallest, one of the smallest heme protein, myoglobin, and more importantly, it's much more well understood, but also very easy to make. And can you utilize as kind of our so-called ligand, the nature's ligand, making models or artificial enzyme to mimic much more complex heme enzymes, who can actually introducing one residue at a time to see what is the minimal amount of structure features to mimic nature, both structurally and functionally. So in comparison to organic molecules, one nice thing about the nature that they're very rigid. So they're allowed to investigate these non-covalent interactions much more readily without the whole ligand become flexible or collapse. So we can actually allow us to investigate these non-covalent interactions much more readily than the organic molecules. And so that's really kind of also they can readily replace with non-canonical or unnatural amino acid and also non-cofactor in that regard. So with that principle, we actually have a program for some years now to really using the native scaffold but smaller proteins to mimic much more complex ones. So this is just a summary of some progress made in my lab in terms of mimicking different metalloproteins. And for example, we have a program that we're published some work in the area of making heme manganese centers. This is the overlay of our artificial system with a native enzyme that can do this so-called lignin degradations to small chemical infused. And also the same kind of center can also be utilized for the bioremediation of aromatic pollutants. We also, this is what I'm going to focus on today, the heme copper center for the OR reactions. And also another one working in making artificial electron transfer for solar energy transfers. And also as a chemist, we are not just satisfied making close mimic to the native enzyme, we also want to go beyond, make airplanes instead of just a bird. And so what we're interested in doing then, we're introducing unnatural or non-canonical amino acids. So can actually go beyond the natural 20 amino acids to have a more functional group beyond that. For example, we're able to replace native methionine with the selenomethionine, oxymethionine, also fluorine-methionine, nor leucine. We can actually isostructurally without changing the structure, major change of active site, but yet we can actually systematically tune the functional activity in the native system. And more importantly, also be able to replace a heme center with also other planar molecule that much more readily to make. Heme center is very costly to make and high energy cost. We're able to actually do two-step synthesis and making metallosalines, replacing that with heme center, and more importantly, can replace iron with manganese and other kind of ruthenium, other type of metal centers So you can actually get a wide variety of activity from the chemical or artificial system into the biological system. And finally, there's so many organic catalysts that have been synthesized and characterized by chemists, but they have not been utilized in the in nature. Only a limited number of metallic proteins. But can we introduce in something like metalloenzyme into the metallocene, I should say, which is a very major class of organic centers into a protein, so can control the solubility and more importantly selectivity, both the chemo, initial, and also other stereo selectivity in this regard. Today, due to time limitation, I'm only going to be able to talk about 3 examples. And so this is the example of interest using myoglobin to mimic heme copper center in cytochrome C oxidase or heme copper oxidase that do the OR. So this is the comparison between the heme copper center and wild type or native myoglobin. And then we can see the differences. Obviously, there's a lack of centers in the above the heme center. They have a similar heme centers, and this one have a copper center with 3 histidines. So the Jeff Sigma in the lab was able to do computer modeling to engineer the heme center by the copper center by replacing the phenylalanine and the leucine here into histidine. He did the computer modeling to show that yes, it can actually generate the heme copper center similarly to the native centers. In fact, we got a structure of crystal structure of it to show yes, actually we can get mimic native one is actually pretty well. Well, this was done some years ago now, just like a state of art at the time. We saw very good structure model, but minimal or no activity. We just said something looks like in nature, but doesn't work like in nature. So what's going on here? So it took a while to actually to read the literature to find out actually, if you look at this one in the native structures, and it's a membrane bound protein, large protein, it's harder to get high resolution crystal structures. So the group in Bloomberg and Seaborn have utilized native crystal structures, be able to using QMM model to be able to hypothesize that the present water and hydrogen bonding network play a key role in stabilizing and polarizing OO bond to do this 4 electron 4 proton reduction from oxygen to water. So this is very, very revealing and insightful into that. But the issue here, in order to turn a structural model into functional model, we have to be able to use introducing water and hydrogen bonding network. So as a chemist, we know how to introduce histidines and other residues, but how to strategically position water into the active site is something that's very difficult to do, right? Especially in the very specific way. And the breakthrough comes through actual recognition that there's actually tyrosine right here that's very critical in the function of the group. And then we recognize that our initial model naively, we didn't actually think about the importance of tyrosine. So Kyle Miner in the lab decided to introducing those features by introducing tyrosine next to the active site. So this is the native wild type myoglobin that again has oxygen bonding site, but no hydrogen bonding network other than this one from distal histidines. By introducing the tyrosine, actually able to introducing, this has got a high resolution crystal structure and able to show that actually we get some water hydrogen bonding network quite similar to the native cytochrome C oxidase. So that's the step number 1. Step number 2, and also we recognize the electron transfer between our model system with a partner cyclone B5, also very, very slow, much slower than the native system. So by both engineering active site introducing hydrogen bonding network involving water, as well as electron transfer, able to in 2015 to show that we actually get our artificial system in myoglobin has activity pretty similar to that of native cyclone oxidase. In fact, we go from almost no turnover into thousands in turnover in this particular regard. In collaboration with Abhishek Dave's lab, we're able to immobilize our model system onto electrode and also compare it into both the native system, cyclone C oxidase, and also the synthetic model. At the time, the state of the art was the Jim Coleman's group, making a very elaborate synthesis of this very beautiful model, the heme copper centers, to demonstrate have a functional, both the structure and function model of native systems. So when we compare the two, we actually show that our model in myoglobin actually about 1 order of magnitude better than the native system, And in the for the second order rate constant, is about 2 order magnitude better than the synthetic model system. So that just shows that having a structural model is not good enough, and we need to model the second spheres. So I want to also show another exciting example of this heme iron sulfur cluster. As I mentioned, there are no reported inorganic catalysts or artificial enzyme to mimicking this very interesting reactions of sulfite reductions. So what we decide to do, and actually Evan in the lab, looking for nature for inspirations. So you look at the crystal structure of native sulfite reductase, or SIR, and he also tried to define the cavity for that can accommodate iron sulfur clusters next to a heme center. So with the inquiry of this cavity, he went into the protein data bank that have all the heme enzymes that has been crystallized and structurally characterized. He's looking for all the heme center that have a similar cavity that can accommodate something like iron sulfur cluster. He able to find out actually cytochrome C peroxidase is one of the top candidates. And so then he using Rosetta, this is the program developed in Dave Baker's lab at the University of Washington, use that to actually be able to define or design 3 of the 4 cysteines that can actually to coordinate to iron sulfur clusters. And then we had using more of an empirical design looking at the computer to do the number 4 cysteines. But in all the line, we actually able to do image minimizations, and then to show that we can get a pretty good primary coordination sphere of the heme iron sulfur cluster, the overlay of a native system in green and our system in pink to be very similar to the native system. Well, when you look at activity for sulfite reduction, we got no activity. Again, similarly, we have something structure looks like, but does not do any function in that regard. That did not discourage Evans from and others from pursuing it, because we know that's not alone enough. So we're actually looking for nature for other sites. So he looked into the second spheres in both the substrate binding site as well as the active site or metal binding site. He actually found out that that in order to stabilize sulfite, which is very difficult to bind, they have to elaborate lysine or arginine that positively charged to be able to stabilize the substrate. But also they're also elaborate hydrogen bonding network, the hydrogen bonding to iron sulfur cluster to tune the redox potential as well as other functional properties iron sulfur clusters. So inspired by nature, and they look into introducing the second coordination sphere interaction. First, the substrate binding site. They're able to introduce lysines and arginine to be able to stabilize the substrate binding site at the similar locations. But more importantly, they're able to also introduce hydrogen bonding network similarly to the native sulfite reductases to be able to actually to tune the property of iron sulfur cluster. So again, I'm going to skip all the structure characterization and spectroscopy studies. The bottom line that the first generation got a minimal activity, and the second generation gets some activity. But the third generation, the combination of substrate binding site as well as hydrogen bonding to the iron sulfur cluster, we're able to get a generation of this cytochrome C peroxidase that have activity above 20% of native sulfite reductase activity. Again, another demonstration that the importance of these weak non-covalent interaction in the second sphere that play the key role in both conferring and also tuning activity. So that's actually one example, 2 of the examples that are important of that. But another example I would like to give is how actually can tune the potential to overcome the overpotentials. Because if you look at literature, many of the fantastic catalysts that can do the reaction similarly to nature, but unfortunately at the high cost of overpotentials. And they drive the reaction at high overpotential in order to meet or to match that of native enzymes. So the nature, on the other hand, they not only carry out the reaction efficiently, but also carry out a reaction at minimal overpotential. For example, I mentioned cytochrome C oxidase is one of the examples. So, well, our question then, can we actually tune the redox potentials or over potential of the artificial enzymes with minimal or no over potential or 0 over potentials? So we start out with an understanding of this so-called type 1 of blue copper centers. It's a very interesting example. So this so-called type 1 blue copper center has been known in many biological systems, including photosynthesis. And so this consists of copper with the 2 histidine and 1 cysteine in a planar or trigonal planar site. So if you look at the nature, the same type 1 copper centers is found in many different biological systems, yet has a very different potentials. Some of them, like a racistocyanine, has about 620 millivolts. Another one, a pseudoazurin or mutant of that, has only 130 millivolts. But if you look at active site, it all have a trigonal site with a copper center involved in there. So the question, the long-standing issue before our work was that, how can the blue copper or type 1 center has almost identical primary portion spheres, but yet wide range of potentials? How does nature be able to vary that so widely? So if we talk in organic chemistry, we can vary the potential in other means, like we change the metal line, change the geometry, change the ligand. The beauty of the challenge for us, the beauty of nature and the challenge for chemists or biologists or for anybody who are interested in the native system is how can you actually change the potential without changing the active site? Because this has been known by many groups to show that this actually is very efficient for electron transfer, because any change of solvent ligand geometry can change electron transfer efficiencies. So nature does not want to do that. They want to change this without changing the primary spheres, but they still can do that. So more importantly, how can we predict the redox potential? When we want high potential, how are we going to do it? When we want low potential, what feature can you do it? And so that's really kind of a question. So we are able to address this question by Nick Marshall and others in the lab. So our idea is that without changing the active site, which is shown here, can we change the second coordinate spheres and then be able to systematically tune the potential without changing the active site? He started out with a native system, azurin, that have potential about in the middle of here, about 300 or 50 millivolts. He able to show that you can change 2 things. One is changing the hydrophobicity. This is measured by the log P, the partition coefficient between oxygen and water, and able to show that when you change it more hydrophobic, you can leucines or hydrophilic asparagine or glycines, glutamines, and they're able to show that you can actually linearly tune the redox potentials with a more hydrophobic or less hydrophobic residues. But unfortunately, can only change that by about 200 millimole range, not about almost 500 millimole range in native system. Then more importantly, they recognize that the hydrobonding network next to the cysteine, according to the copper, play the key roles. So actually look at the nature, looking for the one like racistocyanine have a high potential versus a pseudoazurin with low potential. We recognize it's the number of hydrogen bonding and the strength hydrogen bonding play very important roles. And they can be tuned by 2 mutations. One is the so-called asparagine to serine mutation, another phenylalanine to asparagine mutations. You can see that every time we change that, they actually can change about 140 millivolt. And more importantly, is additive. So we can add hydrophobic mutations, hydrogen bonding of one hydrogen bonding, another hydrogen bonding interactions. So in doing so, we actually take one single azurin that actually make a 1, 2, 3 mutation or 4 mutations. We can get actually variants or mutant of azurin has a potential higher than the highest reported literature for all the type 1 of blue copper centers. Along the same line, by making series mutation to make a more hydrophobic and less hydrogen bonding network, they're able to make the variants or mutant of azurin have a potential lower than the lowest reported literature. This is the line that all the reported potentials before our work that actually had highest or lower potential, we're able to beat out or surpass them by highest and lowest range. But more importantly, now have a series of mutations, we can mix match them. So in doing so, we can actually get a variance of azurin that have a potential of 0 all the way to about 700 or anywhere in between in this particular potential. Again, by just varying 2 parameters, hydrophobicity and hydrogen bonding network. And so that actually get us thinking about how does the biological system is able to tune the potential from both -1, which is the hydrogen is reduced to a proton reduced to hydrogen, and all the way to +1. This is versus SHE, and where the water is oxidized. So our question is, how many protein and mutations does it take for the nature to cover the entire physiological redox potential range from -1 to +1? So if you look at literature from this review articles, we actually reckon nature's answer is a 3 class of metalloprotein and many different mutations. So our idea is that what is the minimum can we use less protein, and what's the minimum mutations to do that? So the answer was done by Parisa in the lab. She able to actually looking at the azurin, we build upon the Nick Marshall's work. They're looking for mutations go beyond 0 to 700 millimole. Can we go to -1 to all the way to +1? The answer was this particular protein. She able to introduce 2 phenylalanine in here to make it even more hydrophobic. He able to make this copper azurin variant has a potential close to 1 volt or about 970 millivolt in this particular regard, again, in water and buffers. And then more importantly, if she replaced the copper center with the nickel, she able to get a variance of nickel azurin has a potential about -950 millivolt. Again, we have 2 metal ions, nickel and copper, several different mutations. We're able to mix match them between 2 metal ions and different mutations. We're able to cover entire physiological network from -1 all the way to +1 and anywhere in between here. So basically able to make water soluble redox agent can be tailored to almost any potential between the 2 range. And also now actually able to tune the, we're in the process, tune the 0 over potentials and also make other oxidation states. So also, as a chemist I mentioned, we're not satisfied with all the 20 natural amino acids. So recognizing that this tyrosine plays a key role, remember that play a critical role in conferring the OR reactions, able to say, can we actually replace this tyrosine with a non-canonical amino acid? Can we make a chlorotyrosine, difluoro or trifluorotyrosines? Why? Because we know from, again, chemical knowledge, when we tune the, change these tyrosine into other type of analog, we can systemically tune the pKa of this phenolic groups. Theoretical prediction to show that the lower the pKa of this tyrosine, the easier to give a proton, the faster the reaction should be. Again, this hypothesis has been done many years ago by others, but it's very difficult to replace a native cyclone C oxidase tyrosine with other non-canonical tyrosines. But we're able to do that in a much smaller system, myoglobin, able to show nicely here by Yang Yu to show that we can actually systemic tuning. By actually lower the pKa, we can get a higher activity. But also more importantly, the product selectivity also improved. We actually get a lower pKa, we actually get more water generation than the native system, and also get another boost in the turnover in this regard. And finally, able Ambika in the lab also able to replace a native heme center with other heme center, because in cyclone oxidase, they can either have a heme B center and heme A center. Depending on what type of heme, they can actually have a very different kind of redox potential and over potential and activities. So she's able to replace a native heme B center myoglobin with either diacyl, monoformal, and diformal species. In doing so, she can systematically tune the over potentials and more importantly activities. So basically, we got even lower potential, even higher activity due to the faster electron transfer and lower potentials. Again, we are on the way to matching our model system in terms of over potential to the native cyclone C oxidase. And finally, I want to use the last maybe 5 to 10 minutes to talk about the work we have carried out in the last year with the bp-ICAM support. So the goal of this particular project is to generate CO2 reduction artificial enzyme to do that. So the way we're able to do them, interested replacing native heme center with the cobaltporphyrin, which is known to be efficient in CO2 reductions. And then we're using the photocatalytic center using ruthenium to be able to drive, using light to drive the reaction to do the CO2 reduction. So what we did, we carried out in the lab by Sas and also Yunling in the lab, able to take out the heme center from the native myoglobin and then replace that with the cobalt. And then again, this is the spectroscopy characterization of that. But more importantly, we're able to demonstrate that we can actually carry out very high turnover, about 1,000 per hour. This actually surpass any artificial enzyme reported so far, usually about a few turnover per hour, by 100-fold. And more importantly, we're able to tune the cell activity by changing the pHs from pH 6 to 7 between the CO2 reduction or CO generation versus hydrogen evolutions. And because again, this is involving some of the protons at pH residue, the pKa of the residues that are important in there. And also recognizing again, second sphere play key roles. If you look at the native center in CO2, carbon monoxide dehydrogenase, the lysine, positive charge residue, and histidine play a key role in tuning that. And also in other preference systems, they also utilize second sphere to tune this particular CO2 reduction activities. So we're actually able to introducing lysines and arginines systematically into the distal side where the CO2 binding occurs. We're able to systematically find out about the role of these kind of a second sphere interactions, we're able to tune the activity even higher than that than the native mutant system. And we'll also recently be able to immobilize these artificial systems onto the electrode and using several different methods, 3 different methods, and then we're able to demonstrate at least on one, actually 2 of the 3 methods, we're able to find the redox couples, and in terms of iron 2, iron 3 couple, and iron 2, iron 1 couples. But more excitingly, we're able to show that we can actually solve the electrocatalytic current due to the CO2 reductions. Again, control with argon, we do not see the electrocatalytic waves. All right, so let me summarize. I think I'm just about the right time to summarize. So I hope they convince you now. It is possible to design complex functional metalloproteins such as heteronuclear metalloenzyme in multi-electron, multi-proton processes using much simpler protein like myoglobin. From this process, we learned that the protein environment around the metal binding site is heterogeneous. Designing metalloprotein taking its non-covalent action in the second torsion sphere, especially water in the social hydrogen bonding network into account, representing the next level challenge. If you're able to succeed in doing so, we will be able to resolve new functional metalloprotein with activity as high as the native enzymes. And some of these action models can even display function even beyond the native protein by tuning the second sphere using the native amino acids, such as, example, using azurin tune the redox potential, also using non-native analog amino acid or cofactor, such as we replacing native heme with a diformal, monoformal diformal hemes. And also, more importantly, by studying this native system, we also provide deeper insight into how biocatalysis works than studying native enzyme, because again, native enzyme sometimes like a membrane bound protein cyclone C oxidases is difficult to study. So we're able to show the role of hydrogen bonding network, the role of tyrosine in the oral reactions, and also in the sulfite reduction cases, we're able to show the role of substrate binding site to stabilize sulfite, as well as how we can tune the property of iron sulfur clusters. In the work supported by bp-ICAM, we're able to have succeeded engineering the photocatalytic cobalt myoglobin that can display the CO2 reduction with the turnover frequencies by 100-fold higher than those other reported artificial enzyme systems. We're able to show that CO2 reduction activity can be enhanced by incorporating hydrogen bonding donors near the active site. And also, we're able to show that we can do not only photocatalytically, but also can do electrocatalytically to do the CO2 reduction using our cobalt myoglobin system. I'd like to thank people who did the work, and particularly the SAS and also Yunling, and also Hui Guang in the lab. But also I'm grateful for the support of bp-ICAM and also the mentors, Dr. Henda, Dr. Yang, for their advices and support. And also thank you for your attention.